What is endomembrane trafficking? Cells are the building blocks of all life. The human body consists of 1 trillion cells, and they are all responsible for carrying out specific functions to keep our bodies running properly. To carry out these functions, cells go through a network of endomembranous trafficking systems between membranous organelles inside the cell. Each of these organelles have their own role in working together to synthesize, modify, store, package, and transport different macromolecules within and out of the cell. In this video, I will be discussing the various cellular products that are transported in and out of the membranous organelles to perform specific tasks for the cell. One of the major processes of endomembrane trafficking is protein synthesis. There are many steps involved in protein synthesis. The process first takes place in the nucleus, known as the control center of the cell. The nucleus contains the majority of the cell's genetic material in the form of DNA that are coiled into chromosomes. The first step is transcription, which is the process of DNA being transcribed to messenger RNA. DNA cannot leave the nucleus, so it must be copied into RNA to be transported within the cell. Transcription starts when the enzyme RNA polymerase attaches to the DNA and unwinds the double helix of nucleotide bases. One strand of DNA serves as a template and is duplicated into an RNA strand. The only difference is mRNA contains the nucleotide base uracil instead of thymine and pairs with adenine as thymine would. Now that messenger RNA is constructed, it can leave the nucleus into the cytoplasm, which is the intracellular fluid outside of the nucleus that contains organelles. The mRNA binds to a membranous organelle called a ribosome, so the process translation can take place and proteins can be synthesized. The sequence of bases on the mRNA form a code for amino acids, which are the building blocks of protein. The triplets of bases called codons on the mRNA pair with the anticodons of the transfer RNA, carrying the associated amino acids attached. The ribosome translates the mRNA codons to bring the base-pairing tRNA carrying the specific amino acids. Once the mRNA is completely decoded and the ribosome has strung specific amino acids together with peptide bonds using the associated tRNA, a polypeptide chain of amino acids is formed, creating a protein. So why do we need proteins? Proteins are a vital part of the human body and play a primary role in many of the body's biological processes. There are many different proteins in the body, and they all have significant functions. They make up your muscles, catalyze chemical reactions, help your immune system fight against foreign substances, move molecules around the body, coordinate specialized bodily functions, and much more. Proteins are essential in making sure the body functions properly. Some other organelles involved in endomembrane trafficking are the different endoplasmic reticulum. There are 2 types of endoplasmic reticulum found inside the cell with 2 different distinct functions. There's the rough endoplasmic reticulum, which has many ribosomes attached to it, giving it the rough appearance. Its primary purpose is to manufacture, assemble, and transport proteins and lysosomal enzymes. The smooth endoplasmic reticulum do not have ribosomes. They are responsible for synthesizing lipids, phospholipids, and steroids, as well as carrying out the metabolism of carbohydrates. The material from the endoplasmic reticulums are transported to different organelles within the cell to continue the system. Another important part of the endomembrane trafficking system is the Golgi apparatus. The Golgi apparatus is an organelle made up of a stack of multiple flattened membranous discs called cisternae. The cisternae carry Golgi enzymes that help with modification of proteins. The function of the Golgi apparatus is to modify, sort, and package macromolecules such as proteins and lipids so they can be transported within the cell or secreted outside of the cell through the plasma membrane in a process called exocytosis. The Golgi apparatus has a cis face in which secretory vesicles from the rough endoplasmic reticulum carrying proteins are transported. The vesicles then fuse with the Golgi membrane and empty their contents into the organelle. The transface of the Golgi apparatus is where the macromolecules packaged in vesicles exit the Golgi apparatus and are shipped to their destinations. Lysosomes are a huge part of the endomembrane system. They are membranous sacs made up of many digestive enzymes used for breaking down and digesting materials that go inside. They are responsible for elimination of waste products, recycling the cell's unwanted material, and digestion of macromolecules including carbs, lipids, and proteins. They maintain homeostasis in the cell by repairing the plasma membrane and engulfing viruses or bacteria that would harm the cell. Lysosomal enzymes are the product of the rough endoplasmic reticulum and Golgi apparatus. The last stage of endomembrane trafficking is the plasma membrane. This is where the macromolecules synthesized and modified through the many processes of endomembrane trafficking are secreted out of the cell. The plasma membrane is responsible for regulating the macromolecules that go in and out of the cell and providing a barrier of protection around the cell, separating it from the extracellular content. As you can see, there are many membranous organelles within the cell that work together in an endomembranous trafficking system to perform specific and important functions that are vital to homeostatic regulation of the body.